Good afternoon, Year 10. So, we're going to carry on with our plant hormones topic now, with a lesson that probably won't take the full 50 minutes, because it's just a short ending to the topic on plant hormones, before we do a little bit of a plant practical next week. Or more accurately, of course, prepare for plant practicals we will do when back in school. Now, the task here again is the same as last time. When we did this task before, I'm not expecting you to write it down again. I'd like you for each one to go through and make sure that you're confident that you know what each hormone does, and for all of the human ones, where they are made. So don't write it down, you wrote it down last time. Just try and make sure that there are none there that confuse you even slightly. Any that you're not entirely certain about, you need to write those ones down. So let's say I didn't know what glucagon was, I'm going to write that one down, because then when I go through the answers you can check if you're right or not. But the ones you're definitely certain about, make sure you've got it in your mind, and make sure your answers are the same as mine when I give them after you've finished pausing me. So pause me now, and we'll move on. Right, ADH is released by the pituitary gland, and it is responsible for making the tubes of the kidney more permeable to water so that less water ends up in the urine, so you save more water for your blood. Testosterone is the male hormone released by the testes, and it causes male puberty and any male secondary sexual characteristic. Estrogen is the female puberty hormone, causes female puberty, every female secondary sexual characteristic, and also grows the uterus lining, and that's released by the ovary, and also by egg cells when they've been released. FSH is follicle stimulating hormone, it's released by the pituitary, and it causes a new egg to develop inside of the ovary. Probably mature is the better word that we should be using. Glucagon is a hormone released by the pancreas, causes glucose to be released from the cells. It causes glycogen to be broken down into glucose. LH is a hormone released by the pituitary that causes an egg to leave the ovary. Thyroxine is released by the thyroid gland, and it maintains a high metabolic basal rate. TSH is released by the pituitary, and it causes thyroxine to be released. And auxin is a plant hormone which causes plants to grow, causes plant cells to elongate, causes plant cells to grow towards the sunlight. Fine, let's have a go at moving on then. We're going to talk about a couple of other plant hormones, and then we're going to talk about a few experiments that we used to prove that plants had hormones. So, now, the first thing I want to do is I want to touch on some more commercial uses of auxins. Now, plant hormones like auxins and similar hormones can be used for several different purposes. Now, we're going to go through some of these, but broadly, there's one that you need to know about in depth, and there's a few others which is just interesting, which might come up as examples in your exam. I just want to touch on them now. And those are things like Agent Orange. Now, routine powders. I'd like you to take down these notes, they are important. So routine powders we're going to learn all about in year 11. We touch on them now because they contain a hormone a lot like auxin, or in fact auxin. So there are several hormones which are a bit like auxin. You can either contain one of those or auxin itself. What happens is if you cut a little bit off of a plant and you dip it in these routine powders, you can then plant that into some soil and it should grow roots. And the point is, these auxins, because they promote cell growth, if you cover the bottom of the plant with it, it makes roots grow. And that means that if I've got a favourite rose bush, let's say, and I want to grow another one, I can just cut a little bit off and I can create a whole new rose bush. That's obviously useful for gardeners, and has some limited commercial uses as well. And finally, if you've got a very rare plant, you can use it to make more copies of that rare plant. Effectively clones. And when we come to cloning in year 11, we're going to talk all about cloning in year 11, we will do a lot more work on routine powders. Fine. Take down those notes if you haven't already, just pause me. Now, the next ones we're going to talk about are weedol and other, basically weed killers. Now, weedol and Agent Orange work in the same kind of a way. Broadly, if you spray a plant with growth hormones, it makes them grow out of control. What happens if they grow out of control is that they eventually run out of sugars and nutrients for growth, and because these auxins are causing them to grow more and more and more, they eventually die. It makes them die. So they'll spring up quite fast and then drop dead almost within a day. The same thing happened with Agent Orange, and this is a more uncomfortable use of these kinds of hormones. This was used in the Vietnam War, and what happened was that these were sprayed all over forests where the Vietnamese fighters, the Viet Cong, were hiding, so that American soldiers could find them more easily. Of course, the issue with destroying whole ranges of forests, firstly, obviously, very, very bad ecologically, and secondly, it causes all kinds of horrible disfigurements in people, these kinds of chemicals when they're sprayed all over their homes. So very, very unpleasant. They both work in the same way. Hormones causing the plant to grow out of control, which causes death of the plant. In this case, trees. In the case of weed oil and other weed killers, usually little dandelions and things like that. So both of those could come up as examples, but the one you need to know about is routine powders. Fine. Now, positives and negatives using plant hormones is something that does come up. So again, you've got some notes here. Unfortunately, today's lesson is a bit note heavy, because there's just some facts we need to know. So, positives of using plant hormones: you can use them to kill weeds, allows them to grow more crops, and allow food to be produced at cheaper prices. That's kind of the key point. And also, in general gardening terms, it's nice to have a garden which isn't full of weeds, and these can be used for that. And then, negatives. Firstly, there's a lot of chemical waste. Secondly, it can be very harmful to wildlife. And thirdly, if used on a large scale, it can destroy plant species which are quite rare. There are a lot of plant species dying out and going extinct. It's not as reported as animal extinctions, because they're not as cute and furry, but it's an issue, and so spraying these everywhere is not necessarily a positive. Fine. Now we're going to touch on two more hormones, and then we're going to finish off by talking about an experiment which was done in order to prove that plants had hormones. So, this is just a brief talk about Agent Orange, but we talked about that already. Some general idea of the bottom right there. These things did cause horrible things to happen to Vietnamese people. Fine. Not fine, obviously. There's two hormones here, which we need to add to that. Gibberellin and ethine. So, gibberellin is a hormone which is useful for making seeds germinate and sprout. So generally, when a seed germinates, when it comes out of the ground, that's because it's got some gibberellin released inside of it. And that's in response to conditions like having enough water around it, being near enough to the surface of the soil, this kind of a thing. So gibberellins are natural hormones, obviously. They make plants germinate. The question then is, we're going to talk about how this could be useful for farmers and for gardeners. Why might that be a useful thing, would you think, is the question. So have a think about that. Why might it be useful to be able to force my seeds to germinate? Well, the answer generally is that if you can force a seed to germinate, you can control the growing period. So, some seeds will only germinate at certain temperatures. If I want them to germinate slightly earlier, if I want them to grow bigger, I can spray them with gibberellin and then they will sprout. Also, it means I can make all of my crops sprout at roughly the same time. Because if I spray them all with gibberellin on a particular day, they will all sprout at the same time, they'll all grow at roughly the same rates. And so when I harvest them, they should all be fully grown. Rather than leaving it to nature, where some of them might be too small, some of them might be overgrown, you're going to spray some gibberellin on them, make them grow at the same rate. So we've got controlling the growing season and controlling when they germinate to ensure that they all germinate at the same time. Those are things you can add to those notes there, useful additions for gibberellin. And then the final hormone, actually the final hormone on this topic, is ethine, and that is there. Now, ethine causes the ripening of fruit. That's not very useful on its own, actually, although in plants it is very useful, but it is useful because we can actually block ethine from being released. and that could stop fruit ripening. Why might I want fruit not to ripen? Why might I want my banana to stay green? Why might that be a good thing? That's the question. What do you think? Let's have a think. And the answer is that most of these fruits are being transported from parts of the world far away. If you think about how long it takes for your banana to ripen and then go kind of black once it's in your cupboard, that would definitely be happening on the ships as it was being transported across the ocean if we didn't inhibit the ethene. So what we do with our bananas and our pineapples and our whatever we're getting from far-flung countries, we spray it with chemicals that inhibit ethene production, and then it is impossible for our fruit to grow ripe. So it'll be green when I pick it, it'll be green when it arrives. And then I can simply not spray the chemicals on them, perhaps give them a wash in order to wash any chemicals off them, and then the ethene will be released naturally by the fruit, and it will start to ripen again. This means again that I can control when my fruit is going to ripen. I can make sure that it's not overripe before it goes on the shelves, because nobody wants to buy a black banana. Fine. Again, add that to your notes. Right. 5 questions here I'd like you to have a go at. I would like these written out in full sentences, so I'd like to make you to make sure that you're completing these in full. They are important questions. So pause me. Right, so what is auxin and what does it do? Auxin is a plant hormone and it causes cell elongation, cell growth in plants, meaning that they will eventually bend towards the sun is the main way we use it. Why do plants grow towards the sun? Because auxin is broken down by sunlight so that the dark side of the plant gets more auxin and grows more, and therefore the plant bends towards the sun. What's the function of gibberellin in a plant? Gibberellin is found in seeds, and it's used to make seeds germinate, to make them sprout. What does ethene do in plants? It causes fruit to ripen. And how can those 3 hormones be used to help farmers grow crops? Well, we can use auxins as rooting powders and weed killers. We can use gibberellin in order to make the seeds sprout at the right times so that they are growing simultaneously and growing when we want them to. Ethene can be used, if we inhibit it, to prevent ripening of fruit. Fine. Now, I think that would be fine. And skip through this task, gentlemen. I think it's very similar. Right. The final thing we need to know about plant hormones is this series of experiments that you see on the board. Now, I am going to talk you through these experiments. And then there's going to be a task which is going to require you to understand these experiments. They are relatively complicated. Once you get your head around them, though, they're not too difficult. So, basically, people wanted to know why plants grow towards the light. It is a strange thing to think about. We tend to think of plants never moving. They just sit there. They might grow. Pretty much the only growth that is very obvious in a particular direction is growth towards sunlight. So that interested scientists. Why did they grow towards sunlight? And we're looking back, as you can see on the picture, towards the 1880s. And we're talking about the Darwin, Charles Darwin. So, very famous scientists involved in this. Now, first things first, Darwin planted himself a little plant, and it's called a coleoptile, but you could use any plant for this. It would be quite a good one. And it grows out of the soil. He shone light from one side, from the left on this picture. Possibly your right, left on my picture. You can see where it's coming from. And you can see that a normal plant, a controlled plant, does grow towards the light. Now he's planting that one just to make sure the experiment actually works. Because if that one doesn't grow towards the light, then there's something wrong with the plants that you have. This just shows that plants do grow towards sunlight, which we all knew. Now there was a theory that it was something to do with the tip of the plant that was making the plant grow towards sunlight. And there was a theory that was something to do with the base of the plant that was growing towards sunlight. Basically, it's the idea of, does the plant grow from the top or does it elongate from the base? I.e., are we just growing new stuff at the bottom, which is making the whole thing push upwards, or are we actually physically growing from the top? And so he did loads of experiments to prove this, one way or another. The first thing he did was that he removed the tip of the plant. He just cut it straight off. Now, what happened there? Well, you can see the plant didn't grow towards the sunlight. What does that tell him? It tells him that the tip of the plant has something to do with the plant growing towards the sunlight. And we know this is because there is a meristem here. We remember meristems from year 9. If you don't remember meristems, they are basically the bits of the plant that have the stem cells, and therefore the bits of the plant that grow. Then he thought, well, is it to do with the tip of the plant just being there, or does the light actually have to hit the tip of the plant? And so what he did was he got basically some tin foil, although it wasn't tin foil at the time. He would do it tin foil today, and he put it on top of the plant. Little hat for the plant that light could not get through. And what happened? Well, the plant didn't grow towards the light. That proved that light had to hit the top of the plant for the plant to grow towards the light. Or did it? Because then there's a thought of, well, maybe plants just don't grow very well if they have hats on. Maybe there's something to do with the top of the plant being compressed slightly, or weight on top of the plant, a little weight from the tin foil. So he had to prove that it wasn't the hat that was causing the problem, it was actually the light that was causing the problem. So then he did another experiment. He got a little transparent cap. We would use cling film today, thin layer of cling film, or maybe even a little perspex kind of plasticky thing. But he got a clear cap, and what happened was it grew towards the light. That proves hats don't stop plants growing. That tells me that with my tin foil hat over here, my opaque hat, that was actually the light that was the issue, i.e. light has to hit the tip of the plant. Then he thought to himself, well, let's prove that it's just the tip of the plant that is the issue. Because the final problem might be, maybe plants don't like tin foil. Maybe tin foil in some way inhibits their growth. So what he did was he got some tin foil and wrapped it around the base of the plant. What happened was the plant grew towards the light. That proved that actually it wasn't the tin foil that was the issue, it was the light. So these 5 experiments together proved that firstly, plants do grow towards light, we already knew that. Secondly, the tip is what is causing the issue here. So remove the tip of the plant, that is what causes the issue. And thirdly, that light needs to hit the tip of the plant, because when you cover it up with something opaque, it doesn't grow towards the light. When you cover it up with something transparent, it does grow towards the light. And so it's the light hitting the tip that is causing the issue. Now, he had no way going forward to prove why this happened. He only proved that the tip of the plant is what needs the light. However, in 1913, they were in a better position to decide what was causing this. So we have a second set of experiments by a couple of scientists called Boysen and Jensen. Now I'm afraid I misspoke. One scientist called Boysen and Jensen. Fine. So this is a very interesting experiment, and this one's quite clever. Broadly, people didn't know now, was this stuff that Darwin had seen caused by a nervous system within a plant, or by some kind of chemical release within the plant? So there were 2 theories. One was that there was actually some kind of electrical signal being sent through the plant, and that just like in a human brain down to the legs or whatever, and that was what was causing this growth towards the sunlight. And the other was that there was some chemical being released. And so what Boysen and Jensen did was he removed the tip of plants, put it to one side, put a little block in the way, and then put the tip back on top. Now what he did was, with one of them, he got a chemical which would allow other chemicals to diffuse through it, and he used that. He actually used gelatin. Now the point of gelatin is it won't allow electricity through, but it will allow other chemicals through. So it's an electrical insulator, but it does allow chemicals through. The other one, he used something called mica. Now that allows electricity through, but it will not allow chemicals through. So, if it had been electricity causing this, then the one here on the right with the mica would have worked just fine, because electricity could still have gone through and the plant would have bended towards the sun. That's fine. But that's not what he saw. So we know for certain, from the fact that it didn't grow towards the sun, that it's got nothing to do with electricity. The one which he did with gelatin did bend towards the sun. And what that told him was there was some chemical which was being released, which was travelling up the plant, and which was causing it to bend towards the sun. Because chemicals can get through, and electricity can't. So, we have 6 experiments here. No, I take about 7 if you include the control. What I need you to do, and you may have to watch me explain that again, or do some research of your own, it's absolutely fine obviously. I'd like you to do 2 things. Firstly, I'd like you to draw this diagram. There's no need to do the roots or anything like that, just for each plant show me what happened. And then secondly, for each experiment, explain what the scientist did is a bit strong, to be honest. As long as you've done your diagram properly, you probably won't need to do that bit, because you will have labelled it what your scientist did. And what it showed. So what is the point of each of these 7 different plants? Each one needs a sentence, and you can lay it out however you like. By the end, you need a piece of work which says, here's what happened in each experiment, here's what it showed. Now, that's going to take you some time. This is the main task for today. And there's a good, I reckon it'll take you at least 10 minutes, if not a little bit longer. If you have any struggles, contact your teacher on Teams. This is the most complicated bit of this small topic, the plant hormones topic. Hopefully I've explained it well enough for you to understand. Any confusions, contact your teacher on Teams, or just go over it online, because there's a lot of research on this in bite size. You can have a look. Loads of websites will have an explanation of this, because it's a very famous experiment. Fine, that will be the end of the task for today, once you've completed that. So, complete that, upload your work, and that will be the end of the lesson. Hope that's been helpful to you. From today, once you've done this task, you should know what gibberellin is, what it does, how we can use it, what ethene is, what it does, how we can use it, how we can use auxins in routine powders and weed killers, and what these experiments are and what they showed. Have a great day.